I mean, I could give a short lecture
Okay.
on how it works,
Yes.
but you gotta pay me.
Yes.
Okay.
We can do that.
Okay.
Okay. So our second lecture, we will talk about is biochemistry, and we're going to do a brief review of every basic knowledge, and we're going to make sure everybody is on the same page. The reason is that when we talk about meat tenderization, for example, we're going to talk about depletion of ATP during the after harvesting. Then we'll talk about glycogen, all those things. Then you don't even confuse about what is ATP is, what is glycogen is, what is glucose is, then that's not very good. So that's why we want to review these biochemistry knowledge before we talk about real food chemistry, then make sure we are at the same page, okay? So this one we're going to talk about is introduction of biochemistry. Now, obviously, biochemistry is very huge, so we're not going to talk about everything. We will focus on aerobic respiration. Now, before we talk about that, we mention about the electron. We talk about octet layers, valence electron, and the sp suborbital layers, all those things. Because all those biochemistry knowledge, some of them could use electron transfer to explain it. So we talk about in the microbiology class the same thing, which is if the electron, we have a donor and acceptor. If this donor is organic, chemical, and the acceptor also organic chemical, we call this is fermentation. And we're not going to spending time talk about today because we will mention this later on when we talk about beer brewing, vinegar manufacturing, and also the wine manufacturing. We'll talk about that. So we want to talk about right here is if the electron donor is organic chemical and the electron acceptor is inorganic chemical. And this is called respiration. Now, it depends on the final electron acceptor. If the final electron acceptor is oxygen, we call it aerobic respiration. And if the final electron acceptor is other chemicals other than oxygen, we call it anaerobic respiration. And we're not going to talk about this one because this is usually for some of the bacteria, let's say sulfur bacteria. They are using sulfate as a final electron acceptor to generate the hydrogen sulfide. So that's for bacteria. And we will talk about here is the aerobic respiration. Now, what is really the final targets for the aerobic respiration is for generate ATP, because this is an energy policy or energy currency. So we're going to introduce you what is ATP looks like very briefly. So ATP, you know, this is called a 5' adenosine triphosphate. So this is what that means. A adenosine, T tri, which means 3, P is phosphate. So this is a food chemistry class. So let's talk about this structure real quick. Okay, what it looks like. Very simple. We have a major one right here. This is maybe too small. I'm going to draw right. Let me see right here. Okay, a little bit. We have this 5-carbon ribose. This 5-carbon ribose is H, OH, H, OH, then you have H, CH2, OH is right here actually. That's a 5-carbon ribose. That's 5-carbon ribose. And you know that we call it DNA deoxyribose, which means this H is gone. Okay, and that is 2 prime. So what we do is 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. And that's a ribose is there. And here we're gonna add a base at the 1 prime position. Now what's gonna be the base? It's gonna be adenine. So what's gonna be the adenine looks like? Is adenine always is a purine. Because it's a purine, so there is 2 different rings. One is 6 carbon, and the other one is 5 carbon. So I have to move this because it's not really fit. So we're gonna have to do right here. This is a 6 carbon, and that is a 5 carbon. Okay, it's straight. Okay, that's the first thing you need to know. Then let's draw it. We draw double bonds right here and here. What we are missing? We're missing the nitrogen because it's important. So we're missing these nitrogen, missing here, and this is H. Okay, so how are you going to label that? Your label is from 1, 2, 3, 4, 5, 6, 7, 8, 9. So that is adenine and connected with 1 prime of the 5-carbon ribose. And then at right here, the 5 prime CH2OH, what will happen is this phosphate will be there. So goes here, then go here. That's 3 phosphates. That is what it means by triphosphate. Okay, so that's why we call it ATP. Now, we also need to know, for a microorganism, or the human body, or plants, during their aerobic respiration, it's going to pump out with GTP. And if you learn about like DNA transcription, translation, they're going to using CTP. And sometimes they use UTP. So you also need to know, not only ATP is the energy currency, ATP, GTP, CTP, UTP, I never heard about TTP, but these also could be a backup plan used as energy currency, as a replacement of the ATP sometimes. So let's talk about these, what the structure looks like, okay? So this is something like we review the purine and the pyrimidine, and we already have the A is right here. So adenine is right here. It obviously is called a purine, one of the purine. Another one of the purine is guanine. So what's a guanine looks like? It's very similar to adenine. So when you draw it, you could be drawn like that, the same thing. 6 carbon and the 5 carbon. Then right here, the 5 carbon are the same. No difference. The difference is right here. Instead of you have an amine group in this 4 prime position, you have a carbonyl group. And here, you added an amino group. This what happened is, so go here, we go here, and then we go, before we go here. Okay, so, sorry. It should be going here. Yes, like that, it's done. So that's guanine. And then, we have pyrimidine. So pyrimidine, I'm just right here. The pyrimidine are different from guanine and adenine is they do not have this 5 carbon sugar, they only have that 6 carbon. So what it looks like, this 6 carbon, and you have N, CH3, H. Okay, you do this. You come here, you go to this guy. The carbonyl is right here. Okay, and this is H. And what is this guy? This is thymine. So what is the difference if you connect it here? The adenine, you put it here, is the 9' position. You connect it to the 5 carbon ribose. But if you have a thymine, this is number 1 position. This guy, you move here. That's the difference because the label is 1', 2', 3', 4', 5', and 6'. Go this direction. Okay, so that's thymine. There is one pyrimidine is very similar to the thymine, but it's not in DNA, it's in RNA, is uracil. C. So what it looks like? 6 carbon, exactly the same. The only difference, instead of you have a methyl group, CH3, you have a hydrogen, which is a proton. So this is, we call it a uracil, and the thymine, the difference is a proton replaces methyl at an unimportant place. Now we have another one, which is cytosine. What's a cytosine looks like? Cytosine is a little bit different, but basically it's still the same. Here are the same. You have a 2H, you have this N, and what you have is you have a carbonyl here. It's the same thing. You have N, and on the top you have NH2, and this is still N, like that. Okay, that's cytosine. So here we let you know is that for this structure, not only ATP is an energy currency, guanine, uracil, and the cytosine, they all could be an energy currency. And we also reviewed about what is DNA and what is RNA. And we know about this is called a base. When you only have a base and a 5 carbon ribose or deoxyribose, we call it a nucleotide. Then connected to phosphates, we call it a nucleoside. Then connected to phosphate, we call it a nucleotide. And when you started to do polymerization, especially you have this 3' hydroxy bonds there, you connect it right here. It's called polymerization, and it becomes nucleic acids. So that's what happens for the genetics. So we briefly reviewed here, then we're going to talk about our aerobic respiration cycle. So let's go 3. So aerobic respiration cycle, what we're going to talk about is we started from glucose, then we go this process, everybody knows, called glycolysis. And generally the product is pyruvate. And the pyruvate, we are going through a transition step, become acyl coenzyme A, and then going through this big cycle is called citric acid cycle, or Krebs cycle, or carboxy acid cycle. And this will generate tons of ATPs. And that is always my homework and assignment in my general microbiology class and also in the food chemistry class, because that is testing you a little bit of math knowledge also there, okay? And then we also talk about these details. We will mention about substrate level phosphorylation, oxidative phosphorylation, how we generate the ATP. Okay, so that's the outline here. Then we're going to draw the detail of all these things. Start from here. Okay, so the camera will be moved when we go the other side. So let's talk about the glycolysis first. Everybody should be very familiar. Let me draw. Okay, we can draw like that. With what is the glucose structure? Okay. H, OH, H, OH, OH, H, go down, OH, H, go here, H, CH2OH. This is glucose. This is the first thing you need to know, because we always going to mention it. Glucose, you write like that, is called a closed ring structure. It could be opened. It could be also write like a chair, called a chair configuration. There's 3 different formats. We will mention later on during the Maillard reaction. Their reaction actually is a little bit different. So here we use a closed ring to talk about that. So glucose, the first step, will add a phosphate at 6th position. Because we talk about 6th position, so you need to know the label is 1 prime, 2 prime position, 3 prime, 4 prime, 5 prime, and the 6 prime. That's on the top. At that moment, we're going to add the phosphates. So what we do is we draw it very simple. Okay, we go here. Others are exactly the same. And you can even copy, if you forgot about it, CH2O, this will be a phosphate. Therefore, this is called glucose 6-phosphate. Now the question will be, where does this phosphate comes from? This phosphate comes from is ATP already existent in the cell structure, hydrolyze, become ADP, and release this phosphate. And then we can talk a little bit more about what's the enzyme for that. And you learned in your biochemistry class, which is using human and animal as a sample, because if it is a eukaryote, so the enzyme is hexokinase. And this is always mentioned, kinase, kinetic enzyme. We can understand about that. But in my class, I talk about the microbiology. I talk about the bacteria. I usually don't talk about this enzyme because for bacteria, they are going through a group translocation. And this rely on phosphoenolpyruvates, PEP. We'll talk about real quick. So there's a little bit of difference between eukaryotes and prokaryotes. Okay, then we have a glucose 6-phosphates. Then what we do? So next step, it is a isomerization process. So that cause, because the name is called glucose phosphoisomerase. Isomerization become fructose 6-phosphate. Now what it looks like? CH2OH, H, H, OH, H, OH, H, COH, H, CH2O phosphate. That's called fructose 6 phosphate. When we have a biochemistry section, we always say the tricky thing for this fructose 6-phosphate is this 5' position is H, is not OH. But all the others is pretty symmetric at the middle. And this is isomerase, which means this is an isomerization process. What means? Their structure, the chemical structure are the same, molecular weight are the same, chemical structure are different, but the components of the chemical atoms are exactly the same. That's called isomerization. So the enzyme called the isomerase. Okay, fructose 6-phosphates go down, will become fructose 1,6-biphosphate. So we're going to do here CHO phosphate, and then go here, you have OH, H, OH, OH, H, goes H, and here you have CH2O phosphate. This is called fructose 1,6-biphosphate. And again, the question, where is that phosphate comes from? That is from, again, ATP, hydrolyzed, become ADP, and the phosphate. Okay, and what's the enzyme? That's called a kinase. That's called fructophosphoryl kinase. So, this is the first steps of a glycolysis, which we are talking about is right here on this top circle, on this top line. Okay, and in this stage, we were using ATP. We did not generate ATP. So we call it investment stage. It's like you put some money, go to the stock markets. You didn't get any benefits yet, but you put maybe $10,000 there to see whether you're going to get something. Okay, that's why it hydrolyzed ATP, become ADP and phosphates. Now we're going to talk about this chain. It's a branch, so we only talk about one side, the other side is exactly the same. This is kind of interesting when you go here. This looks like symmetric. So there is always something going to happen. Is it going to break down here? Cut it. Then become two parts, or exactly the same. So this comes up with CHO, aldehyde, HOH, CH phosphate. This becomes glycerolaldehyde 3-phosphate. Glyceroldehyde 3-phosphates. What is the enzyme for this one? This is 6-carbon sugar, and that is 3-carbon sugar. 3-carbon sugar, we call it aldose. So that's called aldolase. That is the enzyme for that. Glyceroldehyde 3-phosphates. They also exist in a transition step. It is CH2OH, this is called ketone, CH2O phosphate, dihydroxyacetone phosphate, this is called. Dihydroacetone phosphate. Oxyacetone phosphate. Okay. Oxyacetone phosphates. So glyceroldehyde 3-phosphate. Glyceroldehyde 3-phosphates will go a very important step. This step, lots of people will be missing, will be confusing. What they're going to become? COO, add the phosphates there. I'll draw this one, okay, like that. Then we go CHOH, CH2O phosphate. That is called glyceroldehyde 1,3-biphosphate. 1' and 3' biphosphate. Now, where is that phosphate comes from? This phosphate actually is existent in the cell membrane system, so it just come in. And at the same time, what happened is NAD reduced to NADH2. That's called nicotinic adenine dinucleotides. It's an electron carrier reduced to NADH2. And the enzyme is called glyceraldehyde phosphodehydrogenase. Dehydrogenase. Okay, that is a little bit tricky place. There is no ATP generated by that moment. Okay, but there is a phosphate you added. Okay, glyceraldehyde 1,3-biphosphate. When you go down, what happened? COOH, HOH, CH2O phosphate. Glyceraldehyde 1,3-biphosphate, then become glyceraldehyde 3 phosphates. I actually made a mistake on the top. The top one is called, the top one called glyceraldehyde 3-phosphoglycerate. So this one I made a mistake. 1,3-biphosphate. So this is become 3-phosphoglycerate. Okay, this is correct. This one called 3-phosphoglycerate. Okay, what happened here? You have 2 phosphate. When they become 3-phosphoglycerate, you only have 1 phosphate left. What that means? This means you generate ATP. So you transfer to here, then generate ATP. This is the first place. What happened is you invested something, you get something. Well, that's one ATP. So 3-phosphoglycerate. 3-phosphoglycerate go down here. You're going to become COOH, CHO phosphate, CH2OH. That is become 2-phosphoglycerate. Okay, this is the one actually is you just moving the position a position a little bit. 3-phosphoglycerate become 2-phosphoglycerate. So this is called phosphoglycerate mutase. And this is nothing happened, kind of like isomerization, but we don't say it's an isomerase, it is a mutase. Okay, when you go here, what's going to be? Let's just 2-phosphoglycerate. So you go here, you become COOH, CO phosphate, CH2. What's going to happen here? This water will be gone. You're going to lose one water. What is called? Enolase. That's called enolization. That's why this is called phosphoenolpyruvate. This phosphoenolpyruvate is there. We need a kinase. Then become pyruvate. So COOH, C and CH3. This is pyruvate. You have a phosphate here. You have the phosphate here. It's gone. Why it is gone? Because again, ADP connected to the phosphate becomes ATP. So here happened again, ATP generated. So, comes here, we almost can telling you the glycerate is pyruvate generated, and glycolysis is finished. Okay, a little bit messy writing here. So let's talk about something. The question will be, how many ATP generated during glycolysis? And you get the answer is 2 ATPs always. How do you get 2 ATPs? During this stage, we call it an investment. There is a 2 ATP which is hydrolyzed. So we got minus 2 ATPs. And during this second step, the branch section, you're creating 2 ATPs for each branch. So this is 2 ATPs for each branch. And there is 2 branches. So how many generated? 4 ATPs generated. And 4 ATPs. So what's the total? Minus 2 add 4 equals 2 ATPs. And these 2 ATPs generate is 3 substrate level phosphorylation. Now, I want to emphasize that this is what you learned in your biochemistry class. And what's the tricky thing here is, this is saying glycolysis is a closed cycle. It's not connected to the aerobic respiration. If this is connected to aerobic respiration, what will happen? This guy will going through electron transport chain. So there is actually 3 ATPs generated there. And we have 2 lines, so multiple by 2, that equals 6 ATPs. So I always tell people, this is the answer for glycolysis. Should we have 2 different situations? Number 1, if this is only glycolysis, it is 2 ATP generated. If this is part of aerobic respiration, how many ATP generated? It is 2 add 6 equals 8 ATPs. Because that 6 is right there, going through electron transport chain. And we also learned about that only glycolysis, that 2 ATPs, is being cooperated with fermentation process. That's why fermentation is only 2 ATP generated. They only from glycolysis. So there's a remaining question is why bacteria can survive with only 2 ATPs during fermentation? Because they will generate recycled NAD+. Okay? Which means right here, if it's fermentation, this will become lactic acids. And once this lactic acid generates is NADH, oxidized become NAD+. And I always say this guy looks like there's 2 empty space. You could add electrons there. And at the biochemistry standpoint, electron at the same time equals proton from the energy standpoint. Okay? So that is what we call, we get 8 ATPs during part of the aerobic respiration and 2 ATPs only for glycolysis. That's part of the section of the fermentation, but we don't talk about that section today. We also want to mention these 2 ATPs generate is through substrate level phosphorylation. So what this means, substrate level phosphorylation? You have a very easy answer. Lots of you will say, substrate level phosphorylation generates ATPs not through electron transport chain. And I always say this answer is not very complete. This is sometimes I'm asking you, I cannot use this joke anymore. I sometimes ask you, I said, why he's a boy? You tell me because he's not a girl. Which means you just use the opposite answer to replace the correct answer. So then what is really substrate level phosphorylation? This is go back to do this delta G. And this delta G, which is a free energy, we call it the GP's principle, equals delta H, then T and delta S. So that's enthalpy, entropy, and whatever you call it, and it's in a biochemistry class. And this delta G needs to be negative, then the exogonic reaction will be happening. Now, people can do the calculation. They find ATP becomes ADP and the phosphates. They need the energy. How many energy they need? They need a minus 30 kJ per mole. And this is also could be transferred as 30 kJ per mole as a phosphate transfer power. Okay, that's a transfer power. And then the people doing the research. For any of these places, once they happen, phosphate, they calculated the phosphate transfer power. And what they found, the phosphoenolpyruvate, the phosphor transfer power is very high, 61. And here you have 3-phosphoglycerate. That phosphor transfer power is also very high, is 49. And these 2 phosphor transfer power is larger than 30 kJ per mole for ATP itself. Therefore, this reaction is go this way. It goes opposite direction. That's why the ATP is generated. But then you have a question for me. You have a glucose 6-phosphate right here. You have a fructose 6-phosphate here. Why this does not generate ATP? And people do the same calculation. They find the phosphor transfer power of glucose 6-phosphate is only 13. And fructose 6-phosphate is only 9. That's why they cannot let the reaction go opposite direction to generate ATP. Okay, so that is something we talk about, is substrate level phosphorylation. So here we just want to refresh what we talk about in the biochemistry class. Okay, we had pyruvates generate, and we have a phosphoenolpyruvate generate. This will typically go TCA cycle. We'll talk real quick. How about this guy? In the eukaryotes, it doesn't matter. For bacteria, it's going to go back here. It's useful. Because for bacteria, the first step, not to rely on an enzyme, it's rely on group translocation. And that could teach you another 20 minutes, so I'm going to just skip that. Okay, that's called the PEP translocation. So this guy here, for bacteria, is actually important because they go back to help glucose generate glucose 6-phosphate because they don't rely on the enzyme, hexokinase. Okay, then we're going to talk about this citric acid cycle. Citric acid is a circle, so we're going to draw it. So we have COOH, CO, and CH3. This is pyruvate. Okay, pyruvate go a very simple transition step. Rely on NAD reduced to NADH2 again. Rely on acyl-CoA, so generate SCOA. This is acyl-CoA. Lots of the textbook is using SCOA. I want to tell you this is the acronym. So long name, you can go search Google. Very, very long. So we want to make your life easy, we always say SCOA. So acyl-CoA. But if you go biochemistry major, in some of the universities, they will talk about this guy. More complicated. Okay, acyl-CoA will go a circle. So let's talk about that. Since we talk about this is called the citric acid cycle, so we need to know the structure of citric acids. COH, CH2, COH, COOH, CH2, and the COOH. This is citric acids. This is the key. And I always say, you know the structure of citric acid, you can start to do citric acid cycle. Otherwise, you just stuck somewhere. Okay, the next step, I want to explain to you. In my class, for bacteria, citric acid directly become isocitric acids. So I don't make it too complicated, which means this guy moves up. And then go here, COH, H, COOH. Isocitric acid. Like isomerization, when you ever see isomerization, in your biochemistry class, there is another chemical generated, cis-asconate, sometimes called cis-asconic acids. So we just skip that one, okay? It doesn't matter. So isocitric acids. Then we go here, will generate COOH, CH2, CH2, CO, and COOH, alpha ketoglutarate. This has happened. NAD reduced to NADH2 and also release carbon dioxide. So I'm going to circle this guy here. This is important. Alpha ketoglutarate is very activated biochemical ions. It could go lipid cycle, beta reaction, everything. That's alpha ketoglutarate. Then we go this a little bit flat. Become COOH, CH2, CH2, COO, and SCOA. That's called succinyl-CoA. And this happened again, is NAD reduced to NADH2 and release carbon dioxide. So we're going to circle this again. Okay, succinyl-CoA. This is flat. Becomes COOH, CH2, CH2, COOH. This is succinate. Here happened what? Is GDP becomes GTP. That's the reason I want to mention this guy. This is a typical example. The ATP is not here, but they generate an alternative energy currency, which is GTP, is succinate. Going through succinate. Okay, when you see the succinate stuff, The hydrogen ions is at the middle, proton at the middle. That's not very stable. That's very easy to lose. Therefore, it goes up, becomes CH2, CH, sorry, that's COH, and COH. This is called fumarate. And this happened is FAD reduced to FADH2. I'm not going to spend any time to explain this, but you should know, in addition to NAD reduced to NADH2, they have another very important electron carrier, which is found in adenine dinucleotides, is FAD reduced to FADH2. So generate the fumarate. Okay. You see the double bonds right here? That's always gonna put it back. Okay? So what we do? COOH, CH, CH, COOH, which is OH, H2. What is that? Malate. We'll generate malate. And the malate, we're gonna go the last one, which is oxaloacetate. COOH, CO, CH2, and COOH. That is oxaloacetate. Here happened again, which is NAD reduced to NADH2. And this is go back to acyl-CoA. Okay? And that's a procedure. Just want to make sure it is correct. HOH, yes. So that's called the citric acid cycle. Now the key thing is, we want to calculate how many ATPs generate. And where are they? We'll explain to you real quick. The easiest way to do is circle any places which has happened NAD reduced to NADH2.